Hello everybody. So welcome back again. Yesterday we had started a topic about cellular injury and adaptation. So yesterday we completed about adaptation and a little bit part of cellular injury, that is about reversible injury and irreversible injury. So now firstly we will revise what we did yesterday about the reversible injury and irreversible injury. So basically what we have is like this is a question or this is a slide. So this slide is of basically liver. So because here you can see the hepatocytes. So these are all the hepatocytes. So this is a basically a hepatocyte. So this in this slide what you can see is there is an injury. So here you can see an injury is occurring. So here you are seeing an injury. So what you are seeing here in this injury is here you can see that the injury has caused some hydropic changes or there is swelling. So like accumulation of fluid or fat, whatever. So there is some hydropic changes. Those swelling is seen in this middle, and this injury can occur in liver because of some toxic drugs or whatever, any kind of hypoxic injury to liver. So now this injury, what is the scope of this injury or what is happening in this injury? So here you can see whenever this injury is occurring, so basically because of toxic injuries or any kind of shock. So what happens here is there is increase. Obviously we have studied here that whenever there is depletion of oxygen, what will happen? So it's a very simple answer. Whenever there is a depletion of oxygen, so there will be decrease in oxidative phosphorylation. So whenever there is decrease in oxidative phosphorylation, so decrease in oxygen will result in decrease in oxidative phosphorylation, and whenever there is decrease in oxidative phosphorylation, what happen? There is reduction of ATP, and whenever there is reduction of ATP, what this reduction of ATP will do? Obviously it will increase the plasma membrane. Permeability for sodium, and hence this will result in damage of sodium potassium ATPase pump. And when basically sodium ATPase potassium pump, what it will do, it will obviously increase the sodium inside the cell. So this is the cell, so sodium will increase inside the cell. With this H2O will also move. So basically this is the fundament, and obviously this changes with this. Because of that movement you can see these changes, that is hydropic changes. So this hydropic swelling is seen in mostly reversible injury. So this is an example of reversible injury. So I've said everything what's happening in reversible injury. So the main morphological changing which occur in the hydropic swelling, and that hydropic swelling, so the answer to this question will be there is decreased store of intracellular ATP. Yeah, there are decreased store of ATP. Intracytoplasmic rupture of lysosome. So in this injury, this is hydropic swelling only. So this is a reversible injury. So in reversible injury there will be no rupture of lysosome. Again, membrane mitochondrial membrane permeability transition. So MPTP will also not change in this injury. And obviously protein aggregation due to increased cytosolic pH. So cytosolic pH, whenever there is injury, obviously we know that pH of the cell decreases. So this is also high yield point. pH of the cell decreases in case of injury. So this also is wrong. So the option A will be our correct answer for this question. So now coming to another question. Sir, is a 40 years old man. He is pulled from ocean after bloating accident and he is resuscitated. After 6 hours, he develops acute renal failure. So now the kidney biopsy, it gives evidence of karyohexis and karyolysis in renal tubular epithelial cell. So here now the question is giving us some emphasis on acute renal failure. It is saying that there is karyohexis and karyolysis. Now karyohexis and karyolysis, these are the feature of irreversible injury. Now whenever what I said, because in irreversible injury what happened? Irreversible injury, the hallmark of irreversible injury is basically membrane distortion. So there will be membrane breakage, membrane distortion. So as there will be membrane distortion, what happened? Obviously the membrane will break down. So the membrane of this cell have broken down. So what happened? And obviously membrane of lysosome will also break down. Membrane of lysosomes also break down, and all this will be result in what? The cellular component or the cellular enzyme will come out, calcium will obviously get a chance to enter into the cytoplasm, and this calcium will release different enzyme which we have studied phospholipase, proteases, DNAs. So we had yesterday studied that. So because of all those enzyme, endonucleases, so because of all those enzyme, what happened, they will start digesting cellular components, and hence the nucleus will also undergo karyohexis or karyolysis. So karyolysis is just fading of the nucleus, and karyohexis is the nucleus is undergoing fragmentation. So nucleus undergoing fragmentation or fading, the fading is karyolysis and karyohexis is fragmentation. So these all are features of irreversible injury. So what happens in irreversible injury? Now we can say in irreversible injury, what are the biological events? So we know now the injury is irreversible. So the activation of sodium potassium ATPase, decrease in intracellular calcium. So sodium ATPase potassium are not activated, rather ATP, because of decreased ATP the activation decreases. Decrease intracellular calcium, no, calcium increases intracellularly. Decrease intracellular pH, so yeah, intracellular pH decreases. Increase intracellular pH, no, this also none. So the option here is third, that is decrease in intracellular pH. So why pH decrease? Because obviously one, there is breakage of ATP, and hence there is breakage of ATP, AMP will be forming, and this AMP will induce anaerobic respiration. So this will anaerobic glycolysis, and this anaerobic glycolysis will result into formation of lactic acid. This will result in lactic acid. So this will form in lactic acid, and this lactic acid will result in decrease of pH, and hence basically this is also a type of adaptation only. Reduce pH, what reduce pH will do? Reduce pH will help those enzymes which are like proteases, like phospholipases. So they will help those enzymes, and more of injury will be occurring. So now, since this injury, which we are talking about reversible injury and irreversible injury, so this injury, irreversible injury, will result into cell death. So when do we say cell death? So for cell death, we need to have massive caspase activation. We'll talk about this in apoptosis. There should be loss of mitochondrial transmembrane potential. So we'll talk about mitochondrial transmembrane potential. Yesterday also I talked about that in irreversible injury, mitochondrial MPTP. So MPTP decreases. So now the decrease in MPTP obviously will result in decrease in mitochondrial transmembrane potential. So, and that will result in decrease in that ATP formation will decrease. So third is complete permeabilization of mitochondrial outer membrane or expose of phosphatidylserine. So this is all about eat me signal. So what happened? The phosphatidylserine in apoptosis, what happened in the plasma membrane, phosphatidylserine which is inside in the inner membrane will come outside in the outer membrane, and this phosphatidyl in outside and outer membrane is a eat me signal. This is a eat me signal for cells, so that will result in apoptosis. So this is all about apoptosis. Similarly, if there are no such fever, in the absence of clear defined biochemical events, so there are no clear biochemical events are not present, then point of no return or cell death should be considered when any of the one of the following occurs, like cell has lost the integrity of its plasma membrane. So the plasma membrane obviously have irreversible injury. The membrane distortion is the hallmark, membrane breakage is the hallmark. The cell and its nucleus are undergo complete fragmentation in discrete body. So the nucleus has undergone either pycnosis or karyohexis or karyolysis. So anything happened to the nucleus, or the fragments engulfed by, and then there is engulfment of the fragments, and we'll talk about this also, engulfment of the fragments and a form of death, that is autophagy. So now let's come to types of death. So there are different types of death. So basically we will concentrate on apoptosis and necrosis, but there are all many types of death. Like one, autophagy is also a type of death. Pyroptosis is another type of death. Necroptosis is another type of death. Anoikis is another type of death. Mitotic catastrophe is another type of death, and excitotoxicity is another type of death. So there are different types of deaths, and obviously in your Robins you can find these two added. So these two are questions can be asked regarding pyroptosis and necroptosis. and obviously apoptosis and necrosis are very important for this chapter. So coming to necrosis. So what is necrosis? So basically necrosis, it results because of denaturation of intracellular protein and enzymatic digestion of lethally injured cell. So basically necrosis is a type of irreversible injury which we have learned. So irreversible injury. So in this irreversible injury, there is in necrosis irreversible injury, there is cellular swelling. So remember, cellular swelling is feature of injury, except in apoptosis. Apoptosis, the cell doesn't swell, but it becomes smaller, so shrinks. So now there are obviously, so this is a cell. Whenever the plasma membrane is broken, so the internal cytoskeleton is digested by the enzymatic digestion of proteases, because with the help of calcium activating those enzymes. So this will result in enzymatic digestion, and hence necrotic cells are unable to maintain membrane integrity. So we have already said it is unable to maintain membrane integrity, and their contents often leaks out. So because of that leak only, we can say biochemical changes in blood. Yesterday also I have told, because clinically you can correlate whether cell is irreversible injury has occurred or not, and that's why clinicians order different enzymatic reactions tests like LFT, RFT. What do they want to know by all this? Amylase, lipase, they all LDH. So they just want to know whether the reversible injury has occurred or irreversible injury has occurred or not. Basically, another thing we need to know that necrosis occurs in group of cells. So you will have line group of cells. So normally apoptosis occur in one. So like apoptosis is falling off one leaf. So in it actually apoptosis has come from this word only, falling off a leaf. So one single leaf of a tree, falling off a single leaf of a tree, and basically necrosis has come of a group of completely leaves of tree, they fall at a time. So that is necrosis. So obviously in one MCQ this line will be used, group of cells. So now necrosis, basically what happened, the content of the cell is outside the cell. So whenever the content is outside the cell, or the enzymatic reactions are occurring, what happen, obviously body will start the inflammatory reactions. They will recruit neutrophils and all. So neutrophils, macrophages, they will come in the party, and hence inflammation will increase in case of necrosis. So necrosis go side by side with inflammation. So remember, this is also very important point. Necrosis go side by side with inflammation in surrounding tissue. Now enzymes digesting the necrotic cells are derived from obviously those enzymes. There are enzymes, cellular enzymes, which are digesting, and obviously these neutrophils, the inflammatory cells, the leukocytes, which are coming for party or which have come down, they will also secrete enzymes. They will also have digestive enzymes, leukocytic enzymes, lysosomal enzymes, and they will also take part in the digestion of the components of the cell. So in necrosis, in the cells component are digested in two ways. One, the enzymatic lysosomal enzymes, the enzymes secreted by lysosome of the cell itself, and another is the enzymes which are secreted by the lysosomal enzymes which are secreted by the leukocytes. So leukocytes also are helping, and cell itself also helping. Now this concept will help us to explain the coagulative necrosis. Okay, coagulative necrosis, where for some time the structure of the cell is maintained. Now how the structure of the cell is maintained? Because since the blood supply is cut away at once, so the infarct will not get the blood supply, and hence these neutrophils and all, they will not be recruited at once, and since they will not be recruited at once, their enzymes cannot digest that part, and hence for some period of time coagulative necrosis will maintain the outline of the cell. Now in most cell lines, death receptor ligand activate apoptosis rather than necrosis as a default cell death. So remember, necrosis we have talked about, so in most cases, but what happened, death receptor ligand, they will activate apoptosis, not necrosis, as the default cell death pathway. However, if caspase activation in this pathway is hampered, necrotic cell death may occur, acting as a kind of backup cell death pathway. So in most of the cells are designed for programmed cell death. But if programmed cell death cannot occur or caspases can't activate, then in that condition they will obviously occur, necrosis will occur. So what are the mediators involved in the education phase? So now education phase is mostly for apoptosis, but when we say education phase, we can say there are some of the mediators which are responsible for necrotic cell death also, like reactive oxygen species or oxidative stress or reactive oxygen species will result in obviously injury. So we know that oxidative stress which have occurred, it does same thing. It will reduce ATP. When it reduce ATP, obviously it will do same thing, that is ATPase and calcium entry will be increased, sodium and swelling of the cell will occur. The calcium will result in digestion. So same thing will happen. And again, that is calcium. So calcium, calpians, cathepsins and phospholipases. Now these calpians and cathepsins, they are mostly responsible for neuronal death cell, necrotic cells. For in nervous system, mostly calpins and cathepsins act. So calpins and cathepsin act, they results in breakdown of lamins. So they will result breakdown of lamins. In DNA electrophoresis, necrosis column are in. Now this is also very hit question. The figure which we will also see around that in the smearing pattern. So in DNA electrophoresis, necrosis column will be in sparing pattern. Obviously necrosis is not a program form, so the nucleosomes will not be cut at a specific position. Rather it will be position like smear. This is what we say, it will cut like a smear of making a slide. So basically DNA and necrosis column are in smearing pattern. So now let's come to the morphology of necrosis. So in necrosis, obviously we know that there is cellular injury, and that cellular injury will result into the breakage of the membrane, and hence the cellular organ there will be loss of cellular organ. Now we know very well that nucleus and cytoplasm. So obviously cytoplasm will stain pinkish, obviously eosinophilic, and nucleus will stain basophilic. So the nucleus will be staining basophilic and cytoplasm will be staining pinkish. But there are also RNA proteins inside the cytoplasm, and those RNA proteins, what happen, they stain basophilic. Now once organelles are digested, so organelles are digested, what happens, that RNA proteins are also removed by the enzymes, and hence the only left one is the cytoplasm, clear cytoplasm, and that cytoplasm will stain more pinkish. So hence eosinophilic cytoplasm. So there will be increased eosinophilia in case of necrosis. So there is, see, this is a normal kidney. So this is a glomerulus, and these are the cells. So there are different type of cells. So these are the cells here, and this all pinkish what you say is cytoplasm. So here you can see this side, this is the counterpart image of same normal glomeruli, and here you can see there is completely distortion of the architecture is distorted, and here you can see obviously the pinkish cytoplasm is pinkish, and obviously there is loss of the nuclei. So there are loss of nuclei here. So this type of figure, it's defining as necrosis. So this is necrosis, increased eosinophilia, and obviously here you can see, you cannot see through these cells. So there are a lot of things within this cell, but here the ground glass appearance have increased. So the ground glass appearance have increased. So glassy homogeneous appearance will be seen. As a measurement result is loss of glycogen particle. As you say, all the cell components are obviously digested. So because loss of glycogen particles, there will be glassy homogeneous appearance. The cytoplasm becomes vacuolated. Cytoplasm will become vacuolated. So cytoplasm, obviously there will be when something is eaten, so when something is eaten, that space will be created, so that cytoplasm will be vacuolated. So we can say moth eaten pattern. So obviously the enzyme will be digesting that or eating those organelles, and whenever it is eating those organelles, so whichever the part it is getting, so suppose this is the cytoplasm, so suppose this part is eaten, the organelle, this part is, so this will appear like this moth eaten enzymatic digestion because of enzymatic digestion of organelles. So cytoplasm will be vacuolated in case of necrosis. Similarly, as we have talked yesterday also, that in irreversible injury, whenever most of the organelles are digested, the phospholipase of the plasma membrane, they will form myelin figures. So hold phospholipase lipid mass called myelin figures. So they start forming myelin fiber, and that is derived from damaged cell membranes. So myelin figures are derived from damaged cell membrane. So this is another question. And fate of myelin figures. So now myelin figures, the myelin figures, they can either be phagocytosis, they can undergo phagocytosis, or they can also degrade into fatty acid resulting in calcification. So they can undergo calcification. So this figure is showing same thing, light microscopy of tubular, obviously from the kidney only taken. So this obviously you can see all the pinkish area, complete pinkish area, the structure of the cell is not maintained. So here you can see, so there are showing loss of nuclei, there is loss of nuclei and fragmentation of the cells. Ultrastructurally, the mitochondria, there are swelling in the mitochondria. So mitochondria have undergone swelling of the mitochondria, and obviously because of phospholipase, this myelin shapes can be seen. So myelin figures can be seen. So myelin figures, they are derived from the damaged cell membrane, holes of laminated phospholipids and calcium if they undergo calcification, saponification. So nucleuses, so in nucleus also irreversible changes, or in necrosis will show different changes, that is it will show karyolysis, karyohexis. Remember, pycnosis will be shown by apoptosis. In apoptosis you can see pycnosis. So this is all about nuclear pycnosis. So the nucleus have gone like shrinkage of the nucleus. Karyohexis, there is fragmentation of the nucleus, 1 2 3 4. So different nucleus have been broken down, like here you can see, here you can see, here you can see. So these are fragmentation, karyohexis. And karyolysis is fading of the nucleus. The nucleus undergo, like you can see this nucleus, it's undergoing fading. So this nucleus is clear one, but here this nucleus, they are undergoing fading. That is less basophilic. They are becoming less basophilic. Nucleus is more is basophilic, so it's becoming less basophilic. So that is karyolysis. So electromicroscopic changes also you can see, as we know, we have yesterday learned changes in irreversible changes. First change is biochemical change. Second is the ultrastructure change. The third one is the light microscope, and finally there will be cross. So obviously ultrastructure changes you will see by electron microscope, and electron microscope, in that changes what you will see, there will be discontinuation in the plasma. Obviously what you will see, so this will be the cell, and when and they are the microvilli, so there will be loss of microvilli, microvilli will be loose, and hence discontinuation of plasma membrane and the organelle membrane. So organelle membrane and plasma membrane, they will discontinue. Marked dilation of mitochondria with appearance of large amorphous densities. So obviously in mitochondria large amorphous density will accumulate. And intracytoplasmic myelin figures. So myelin figures, intracytoplasmic myelin figures will increase with amorphous density and aggregation of floppy material, probably represented by denatured protein. Obviously when the pH decreases, so with decrease of pH, denaturation of the protein will also occur. So with decrease of the pH, denaturation of the protein also occur, and other also enzymatic denaturation is also occurring. So now this figure sum up our necrosis. Necrosis will be somewhere. We have a normal cell. So this is a normal cell with rough endoplasmic reticulum and with ribosome attached to those, and also we have mitochondria and all different organelles. Now whenever there is an reversible injury, so in reversible injury what we can see, there will be swelling of rough endoplasmic reticulum, and obviously the ribosomes will detach from it, and there is also swelling of mitochondria. So swelling of endoplasmic reticulum and mitochondria, and because of swelling you can see valves also forming, and slowly if the injury progresses to them, then we can see breakage of the membrane. So there is leakage of the content will occur from this. Leakage of the content will occur. So breakage of the plasma membrane and organelles and nucleus leaks, and obviously there will be amorphous densities within mitochondria, and inflammatory cells will come, and obviously myelin figure will be seen. So this sum ups our topic necrosis. Now there can be different types of necrosis. So first one is coagulative necrosis. So this is the most common type of necrosis, and obviously occur because of hypoxia, where except in brain hypoxial injury will result into liquefactive necrosis. And coagulative necrosis, basically we know that already I've talked, it will the outline of the cell will be preserved for some days, obviously because the enzyme within the cell will start digesting those cells, though the blood supply had reduced, so leukocytic enzyme are not coming, but within the cell will start digesting it there. And obviously there will be the structure will be formed as the structural protein enzyme are also denatured, hence proteolysis is blocked. So as I already heard, proteolysis is blocked in this case, and obviously feature will be that of necrosis, where is eosinophilic cytoplasm, glassy appearance of the cytoplasm, loss of the nuclei, or we can say karyohexis and karyolysis of the nuclei. So all the features will be seen in local area of coagulative necrosis. And remember, local area of coagulative necrosis can occur in a local area, and that is called infarct, which is wedge shaped, mostly wedge shaped. So this infarct. Now coming to infarct, so there can be white infarct or red infarct. So white infarct is also known as pale or anemic infarct, that is white infarct is blood is not reaching there. So seen in most solid organs with single blood supply like heart, kidney, spleen, so and arteries. So there's single blood supply and arteries, they will be giving white infarct. There is no place for other for blood to come over there. Red infarct mostly hemorrhagic infarct. So the infarction will occur, but there is a lot of blood coming from different supply, like seen in spongy organs, lung, or organ with dual supply like lungs and liver. Liver in hypovolemic shows white infarct, but liver in hypovolemic shock will show white infarct. Remember this. Organ with collaterals like intestines or torsion of testes and ovaries. So this all will result into red infarct. But this is a high yield point, that liver in case of hypovolemic, if there is in the question, read it properly, if there is hypovolemic shock written and the organ is liver, then it will show white infarct. So these are very easy questions which can be asked, a direct question. Another example of coagulative necrosis is dry gangrene and Zenker's degeneration. Now remember, you can be asked about this Zenker's degeneration. You can be given option Zenker's degeneration, dry gangrene, red gangrene, and obviously brain infarct. So the option will be, best option will be dry Zenker's degeneration. So this question can be also asked, which of the following shows coagulative necrosis. So next type of necrosis is liquefactive necrosis. So liquefactive necrosis have digestion of dead cell transforming tissue in liquid or viscous mass. Obviously liquefactive necrosis, enzymatic reaction are there, blood supply is well, so the enzymatic reaction will occur, and those enzymatic reaction will result into autolysis and heterolysis. So the ends, because of the cavity, mostly in the enzymatic reaction or liquefactive reaction, cavity will form. You can see a cavity. So like abscess cavity, bacteria, because of bacteria, so mostly occur because of bacteria and fungus. And obviously hypoxia to brain. Now this mechanism is not understood very well, but hypoxia to brain causes liquefactive necrosis, not coagulative necrosis. So this is obviously showing a cavity, and obviously this is an example of liquefactive necrosis. And obviously this figure is showing that the tissue is, there is loss of architecture. So there is obviously in coagulative necrosis architecture is maintained, but here is loss of architecture is lost, and so this is because of dissolution of the tissue have undergone dissolution in this case. So this is also liquefactive necrosis. Another is gangrenous necrosis. This is a special type of necrosis. It's a non-specific pattern of cell death and mostly applied to the limb that has lost blood supply and undergo necrosis. So firstly it will the dry gangrene will show coagulative necrosis, and obviously bacterial infection superimposed, then it will undergo liquefactive necrosis, that is in wet gangrene. cages necrosis. So cages necrosis, sometimes very easy question comes. Tuberculosis shows which type of necrosis, and we do mistake, sometimes we do mistake, that silly mistake, coagulative necrosis, cases necrosis. Very simple question, you need to understand. It's the foci of tuberculosis infection will show cases necrosis. So this will appear like cheese like friable white appearance. So it will appear like cheese like white friable appearance. So cheese like friable white appearance, so like cheese. So sometimes you will say that cheese like appearance is seen in this infection. What is that? So you need to know about this word also, cheese like. So cheese like is seen in cages necrosis. So they are obviously, cages necrosis is also structureless collection of fragmented or lithe cell. So in cages necrosis they are obviously lithe cell and amorphous granular debris within inflammatory border, granuloma. So we know granuloma, collection of different inflammatory cells, histocytes, and like histocytes like sleeper like histocytes, and all those forms granuloma, and within that granuloma there will be collection of this cheese like material, that is cages necrosis. So here this figure is a very hit figure. So here this comes as cages necrosis, and this can be asked in exam. It has been asked also, yellow white cheesy debris. Fat necrosis, so is a focal area of fat destruction actually. So it's more than of a necrosis, it's a fat destruction. So there will be chalky white appearance, chalky white areas. So now here you can see again, because of fat saponification there will be chalky white area, shadow or outline of shadowy outline of necrotic fat cell. So there will be necrotic fat cell, there will be shadowing outline with basophilic calcification deposit over there. Mostly occur in so frequently asked question is pancreatitis. In pancreas it occurs. Obviously breast also, when sometime injury to breast, direct injury to breast, someone like two girls are fighting or two people are fighting and a gets someone gets punch on the breast. So in that time also you get fat necrosis. That's a very clinical important clinical scenario, or where the sentence, the way you get question, the trauma to breast and obviously subcutaneous tissue. So this are showing fat necrosis, but basically very high yield point is pancreas. Fibrinoid necrosis, so it is immune reaction, because of immune reaction in the blood vessels. Antigen antibody complex will result in this fibrinoid necrosis. So this will show pinkish area, complete pinkish area. So like this, so these are all fibrinoid necrosis. So the answer to this question will be fibrinoid necrosis. So the example of fibrinoid necrosis, like in case of malignant hypertension, in vasculitis, rheumatic heart diseases like Ashoff nodules in central fibrinoid necrosis, peptic ulcer in autoimmune diseases, or SLE, RA, there you can see fibrinoid necrosis. So with that we complete about necrosis. So in necrosis there are some of the examples of necrosis, like reef of fusion injury. So ischemia, reef of fusion injury. So in ischemia, reef of fusion injury you can get necrosis. That's a very wonderful example of necrosis. So whenever there is injury to a cell, now if reef of fusion is, you are again establishing blood supply to that organ or to that cell. So whenever there is a resupply of the blood supply, what happens with that, you are giving oxygen supply to that organ, but that organ is not working properly. The mitochondria of that organ is not working properly. So whenever the oxygen goes, it cannot completely use that oxygen. So free radicals will be generated. And whenever the free radicals will be generated, we know reactive oxygen species are, or free radicals are one of the executioner of necrosis. So they will execute necrosis. So that will result in necrosis. Obviously again there is your defense mechanism will also not work during that time because of because of necrotic cells. So that will also result in necrosis. Another example of another way of this, with blood supply there will be increase of calcium. Obviously calcium will also enter with that blood supply, and where there is already injury in the cell, and because of reperfusion calcium increases, and because of that calcium what happens, again MPTP will open, and whenever MPTP will open, that will again change the mitochondrial conductance, and hence again injury, same process will follow. No ATP, sodium, potassium, calcium entry again, and all lyse of the enzymes, and same process will again follow. Similarly, there is inflammation also. Inflammation can also result in basically inflammation is an also example of inflammation is also can also result into necrosis because of reperfusion injury, because with inflammation lot of obviously where there is injury there will be lot of inflammatory cell, and dead cell is a signal, obviously the signal for inflammatory mediators. So inflammatory mediator, dead cell will obviously call those inflammatory mediators, and obviously cytokines, different cytokines which are released because of inflammatory cells which were recruited over there, they will also activate necrosis. So this inflammation will also result in necrosis, and similarly activation of complement system can also occur in case of blood reperfusion, because of some unknown reason what happened that necrotic cells have IgM IgG antibody stuck to it. So whenever blood supply again restarts in that organ, what happens, those IgM IgG gets activated, and they activate complement system, and that activation of complement system will result into necrosis. So this ischemia reperfusion injury is a very good example of what we call it necrosis. Similarly, there can occur some chemical injuries. So what are these chemical injuries? So like some of the direct chemical injuries or indirect chemical injuries. So direct chemical injuries because of mercury poisoning, you can get direct chemical injury. So what these direct chemical injuries do? So they will again result into injury to the cell membrane, and obviously change the membrane potential. Whenever the membrane potential is changing, a reduce in ATP, and same cycle will follow. And obviously indirect injury is like conversion of those components into some toxic agents like CCl4, which is converted into CCl3 free radical, that will result into injury, and that will result into necrosis. So these are some of the example of necrosis. So ischemia reperfusion injury or direct injury. So chemical injury is an example of necrosis, and ischemia reperfusion injury is also an example of necrosis. Now let's come to next type of cell death, that is apoptosis. So apoptosis is another type of cell death which occurs. So they are basically internally programmed series of events occur, and hence they are more focused on DNA material, that is gene material. So they are gene product, they are more dedicated to gene product. So cell, they breaks into fragments and on apoptotic bodies, and hence those are cell breaks of an apoptotic bodies are formed in apoptosis. So in this, remember, plasma membrane will remain intact. So plasma membrane, it will remain intact, but structure will be altered. Targets, structure, they will be obviously altered, but plasma membrane is intact. So we know that shrinking, cell size decreases, so shrinking occurs in that. So whenever there is shrinking, obviously the structure will be altered, but target for phagocytosis will be target, and obviously that shrinkage cell will be target for phagocytosis. And remember, it will not elicit any kind of inflammatory reaction. So there will be no inflammatory reaction. In necrosis there was inflammatory reaction, but here is no inflammatory reaction. In apoptosis there is shrinkage of the cell, but in necrosis there was increase in the size of the cell. So these are very important difference between two types of reaction. Now apoptosis, it can occur in physiological situations. Obviously physiological apoptosis can be physiological and pathological, and necrosis is mostly or always pathological. So necrosis is always pathological. Apoptosis can be physiological, like your formation of free independent digits. So sometime because of apoptosis only, the 6 digits are converted into 5th digits, and this wave is also removed because of apoptosis only. Similarly, development of brain also requires apoptosis, obviously, and there is development of reproductive organs. We know that Mullerian factors they are inhibited, so Mullerian inhibiting factors, and hence the male genitals are formed. So male organs are also formed because of that. Similarly, there are in proliferative cell population like B lymphocytes, obviously reactive lymphocytes will be removed from those cells, harmful elimination of harmful cell-free lymphocytes. So this will all occur by physiological situation. So these are some of the example of physiological apoptosis or physiological apoptosis. So in pathological situations, obviously apoptosis elements cells that are genetically altered or injured beyond repair. So those cells which we cannot repair, and now that cell is in a program manner, yes, now you need to die because you are of no use to us. So and you need to die. So that in that condition, that is called program cell apoptosis. So this pathological situation, apoptosis occurs without eliciting a severe host reaction, like death of Amitabh Bachchan in Sole. That is an apoptosis. Example of apoptosis is a very programmed cell death, because he knew that he had got injury and now he can't save to save other cells like Dharmender and Hema Malini. He died himself. So that is example. So in a very programmed manner, death is all about apoptosis, and necrosis is all about in a very random sequence, in a very randomly someone is coming and just firing around everybody. So there is necrosis, a very random necrosis. Necrosis is random. So what happened, like DNA damage, another is accumulation of misfolded proteins. So misfolded proteins accumulation, different accumulation of, like in Alzheimer's disease, in cystic fibrosis, CFTR, Alzheimer's A beta proteins, those accumulations are, or Parkinson disease, synuclein also is a result of this synuclein, that also can result in apoptosis. So cell injury in certain infections, in amyotrophic lateral sclerosis also there is so because of apoptosis, this some of the disease are also occurring. Now pathological atrophy in parenchymal affected after duct obstruction, obviously because of duct obstruction in some of the parenchymal organ, this can occur. So these are the pathological situations. So what is the morphological changes in apoptosis? So there is shrinkage of cells. So in pathological changes, shrinkage of cell, we know that very well. In necrosis, obviously there will be increase in size, but in this there is shrinkage of the cell. There is condensation of nuclear chromatin peripherally under nuclear membrane and cytoplasmic condensation. So we know that there is condensation of nuclear chromatin or pycnosis. What we want to say is pycnosis, nuclear pycnosis will be occurring. Nuclear chromatin will condense and hence it will form like hyperchromatic, more of hyperchromatic. And cell breaks down into membrane, and obviously cell will break down into membrane. So when this cell, obviously when this cell undergo apoptosis, so it will break down in small apoptotic bodies or vesicles with intact cytoplasmic organelles with or without nuclear component. So nuclear component might be present in them, but cytoplasmic organelles, they are intact. So and in case of necrosis, cytoplasmic organelles are digested, they are not intact. So these cytoplasmic organelles are not intact on those cases. Again, unlike necrosis, apoptosis is not accompanied by inflammatory reaction. So there is no inflammatory reaction. And phagocytosis of apoptic body by adjacent healthy cell in epithelia, or they may be sloshed off by or phagocyted by macrophages. So this also we know eat me signal will be generated by a apoptotic cell, and as I said, the phosphatidylserine, as I already said, the phosphatidylserine will move towards outside on the outer membrane, and that will give eat me signal, and that eat me signal will help for the adjacent cell to eat it, or some macrophages will phagocyte it. So this is example of an apoptotic body. So obviously apoptotic body will be seen in apoptosis. So morphological figure, apoptotic body. Similarly, the best thing or best biochemical event which occurs in this is DNA breakdown or cleavage of DNA, and we know that endonuclease will break it into the fragments of 180 to 200 base pairs, and obviously it will show step ladder pattern, neither smearing pattern will be shown in necrosis. So necrosis will show smearing pattern and it will show step ladder pattern. So DNA and protein breakdown will show step ladder pattern. Here, typically ladder pattern it will be shown by agarose gel electrophoresis. In necrosis, DNA background is random. Obviously, because of that randomness, it's like smearing of a slide, smearing of the sample on a slide. So in a slide, you can smear that like. So now this is a very hit question and it has been asked in AIIMS also. This one is normal. So this one is normal case, and this obviously this is showing case of apoptosis because there is a ladder like pattern in this, and this here it's a smear pattern, completely smear pattern, and this is of necrosis. So C is cell necrosis, diffuse smearing pattern, and B is ladder pattern, and obviously viable A are the viable cells. So it's in this electrophoresis. So this is also another example. Which of the following cellular process best accounts for the presence of scattered acidophilic body in the liver biopsy? So in the liver biopsy, which of the cell will show acidophilic bodies in this liver biopsy? So here, this aggregation of intermediate filaments. Okay, no, intermediate filaments will not show this type of appearance. Coagulative necrosis will not show. Coagulative necrosis completely out, structure of the cell will go. Intranuclear viral inclusion will obviously eosinophilia will increase, but apoptotic bodies will not be shown. So apoptotic cell death is the best account of presence of acidophilic bodies in this liver. Now this figure will also sum up apoptosis. So what are this? Again a normal cell, and obviously this normal cell, what are they? There is condensation of chromatin. Chromatin will undergo condensation and it will become compact. So hence the nucleus will look like pycnotic nucleus, and there will be blebbing of the membrane. Membrane will start blebbing. So in apoptosis also you can see membrane blebs forming, and also in reversible injury. And obviously those blebs will start forming apoptotic bodies, and those apoptotic bodies will later on will be engulfed by AIIMS signal, which will be presented by those apoptotic bodies, and those AIIMS signals will result into phagocytosis. So apoptotic body will undergo phagocytosis, whereas necrotic cells undergo lysis, or they are undergoing lysis or digested by the enzymes. So now let the very important topic, how apoptosis occurs. So apoptosis, the fundamental events in apoptosis is activation of enzyme called caspases. So basically in apoptosis, most important thing which is occurring is activation of caspases. So caspases are cysteine dependent aspartate specific proteases. So these are cysteine dependent aspartate protein divisions. So caspases, C for cysteine proteases, and aspase is lysis or breakage. So they will result in breakage. So activation of caspase, member of family of cysteine proteases, and they divide functionally into two groups. So caspases can be initiator caspases and executioner caspases. So we can have a simple story like a story. Okay, when we talk about story, we'll talk in we can talk in Hindi also. That will be better. It will build more bond. So now caspases का क्या था? ये इसकी जो कहानी है बहुत लंबे उधर से आती है. लाइक द फर्स्ट कैस्पेसेस जो बना था उसको सेड 3 बोलते थे. सेड 3 सी एलिगेंस से आया, क्लोस्ट्रिडियम एलिगेंस से आया सी से. और तब क्या धारणा बन गई कि नॉर्मली जो कैस्पेसेस होते हैं ये जैसे ड्रोसोफिला में बाद में पता चला, ड्रोसोफिला में 7 टाइप के कैस्पेसेस मिले और सेड में सिर्फ एक टाइप का मिला, तो सी एलिगेंस में. सो ना तो क्या हुआ कि ये था कि जितना ऑर्गेनिज्म डेवलप्ड उतना ज़्यादा कैस्पेसेस, जैसे कि ह्यूमंस में अभी 11 कैस्पेसेस हैं. सो ह्यूमंस में अभी 11 कैस्पेसेस हैं. देयर आर 11 कैस्पेसेस इन ह्यूमंस, and the first caspases was developed was SAID3. SAID3 was first caspases developed. Now basically not so important information, but obviously this can be asked somewhere in exam. You can get somewhere in exam that which one of the first caspase developed, or how many caspases are in human. So 7, 6, 10, 11. So it's a very easy question, and hence this can be asked in exam. Again, what we need to know is but बाद में क्या पता चला कि जो सी एनिमल्स में है, सी एनिमल्स में ह्यूमन से भी ज्यादा लगभग, सॉरी सी एनिमल्स में लगभग 10 क्या स्पीसीज निकल गए. सो इट्स नॉट, और सी एनिमल्स प्रिमिटिव ऑर्गेनिज्म है. सो ये जरूरी नहीं है कि क्या स्पीसीज, ये थ्योरी लोगों की फेल हो गई. So this was only simple story. So now let's come back to the topic. So there are initiator caste species and executioner caste species. So now initiator caste species are like 8, 10 and 9. Caste species 9, caste species 8 and caste species 10. And executioner caste species are 3, 6 and even 7. 7 is also executioner caste species. Now remember, they exist as inactive proenzymes or proenzymes. So they exist as proenzymes or zymogens and then undergo enzymatic cleavage. So zymogen enzyme is the first regulator, first natural regulator of caste species. So you can be asked, the zymogen enzymes zymogen, that is the first natural regulator. So there are different types of regulator for caste species, we will be learning that, but zymogen enzyme is the natural regulator of any what we call it, caste species. So basically zymogen enzyme is the first regulator. Now that should be understood very well in this. Now the presence of cleaved active caste species is a marker for cell undergoing apoptosis. So how do you know cell is undergoing? So we presence of cleaved or active caste species. So we see caste species and we know that this cell is undergoing apoptosis. So for apoptosis, caste species, cleaved caste species are the active marker for cell undergoing caste species. So again, I've talked already this, that there will be membrane alternation and recognition by phagocytosis in apoptosis. So there will be change in the plasma membrane of apoptotic cell that will promote recognition of dead cell by phagocytosis. How? Phosphatidylserine will move outside the in the outer membrane, and hence this will be recognized by number of receptors on the phagocytes, and hence they will generate an eat me signal and they will be phagocytes. So phosphatidylserine is moving outside. Very important. I am saying it again and again, phosphatidylserine, but because we'll add one more point to this line, whenever because of movement of phosphatidylserine in outside cell membrane, we'll add one more line. So this obviously, this movement of some phosphatidylserine, notably phosphatidylserine, from inner leaflet to outer leaflet of membrane is recognized by number of receptors on phagocyte. Now these lipids are detected by a binding protein called annexin 5. Thus annexin 5 staining is commonly used to identify apoptotic cell. So this point can be an MCQ. Annexin 5 staining is commonly used to identify apoptotic cell. But is annexin 5 based for apoptosis? No, annexin 5 is not based for apoptosis. Annexin 5 is not based for apoptosis because why? See, obviously phosphatidylserine is inside in the inner membrane, and that will come out to the outer membrane, and that will be detected by annexin 5, and we will know that there is occurrence of apoptosis. But in necrosis also there is dissolution of cell membrane, and hence when there is dissolution of cell membrane, obviously necrotic cell will also stain annexin 5. Yeah, it will also stain annexin 5, though there is dissolution of membrane, so that will not stain like that of apoptosis, but yes, that will stain by annexin 5. So now how do you differentiate? So then you use propydium iodine. So now double positive, so we call it double positive by propydium iodine. So propydium iodine, so this propydium iodine, it stands only obviously necrotic cells. So propydium iodine, so annexin 5 and propydium iodine positive at once will result into necrotic cell, and we can stain by propydium iodine. Staining can be done to for what we call it, this apoptotic cell. So annexin 5 can be a question asked in exam. So we are told again, there are a mechanism of apoptosis. It can be divided into two phases, initiation phase and execution phase. So in initiation phase, that is initiation caste species will act, and in execution phase, executioner caste species will act. So obviously in initiation phase, caspases will become catalytically active, and obviously this catalytical active caspases will act on the caspases education caspases, and they will trigger the degradation of critical cellular component. So this two phase combined forms a proper mechanism of apoptosis. So obviously initiation phase occurs in two ways, that is intrinsic or mitochondrial pathway, or extrinsic or death receptor pathway. So there are two pathways, intrinsic mitochondrial pathway or extrinsic death receptor pathway. So mitochondrial pathway, that is intrinsic pathway, and the extrinsic death receptor pathway, that is extrinsic pathway. There are two distinct pathways where caspases activation occurs. So here the intrinsic mitochondrial pathway. So this figure here, whenever there is a normal cell which binds to a receptor. So remember, again I am saying, when you talk about receptors, please learn about the receptors, survival signal receptors. Learn about those receptors like AKT pathway, PI3 AKT pathway, or GPCR pathway, and learn about RAS and MAP kinase pathway. So you please read side by side, read your physiology and biochemistry also side by side. That will help you understand this topic also and that topic also more properly. So remember, cytochrome C, they are on the inner membrane. So these are all the cytochrome C. So this cytochrome C, for not releasing of the cytochrome C, there are some of the anti-apoptotic factors like BCL2 and BCLX. So these non-apoptotic factors, so these anti-apoptotic factors BCL2 and BCL, will they keep this cytochrome C inside the inside membrane, and hence there will be no leakage of cytochrome C in the normal cell, in the condition of normal cell. So whenever there is normal mechanism is occurring. So now in case of apoptosis, when there is lack of survival signal, so lack of survival signal, that I am saying lack of survival signal, like lack of those PI3 AKT pathways, and also obviously that lack of those pathways, what happens, and obviously direct irradiation or direct DNA damage, what I will do, they will activate the sensors. So remember, there are three things which we need to know, that is anti-apoptotic, pro-apoptotic, and sensors. So there are three things: anti-apoptotic, pro-apoptotic. Pro-apoptotics, they can be sensor also, third type, that is sensor, and BS3 only. Remember, the sensor are normally BS3 only protein. So normally anti-apoptotic proteins and apoptotic proteins, pro-apoptotic proteins, they have BH1 to 4, but sensors, they have only BS3 protein sensors. So they have only BS3, sensors have BS3. So there are three things which we need to know. Yes, here this figure will say BCL2 family. So a large family of 30 members which is identified and belongs to both. So there are anti-apoptotic factors and there are pro-apoptotic. So remember, pro-apoptotic, these are sensor. So which are the sensor? So remember, anti-apoptotic have BH1, BH2 and BH3 proteins. Pro-apoptotic has back and BOC. So they are also BAX, BOC and BOC are BAX, BAC and BOC. They are pro-apoptotic, and obviously these are sensors. These are activated first. So sensors are, for you can remember this way: BAD, VIM, BID for Noxa and Puma. So these are very important sensors. BAD, VIM. So your Mahabharata, go to the BAD, VIM, bid for. So he is bidding for Puma and Noxa. So BAD, VIM, bid for Puma and Noxa. So this will help you remember that they are sensors, and these sensors, they are activated first in case of apoptosis. So activation of sensor, that is BS3 only protein, BAD, VIM, BID, Puma and Noxa. So any of that protein are activated. So these are they will result in antagonism of BCL2. BCL2 is anti-apoptotic factor. So BCL2 is anti-apoptotic factor. Then or BCLX also, BCL2 or BCLX. So BCL2 or BCLX, BCLW. So you need to remember BCL2, BCLX and MCL1. So these are three important ones which you must remember. Now, so what happened? So here, hence the cytochrome C will be releasing out. Now cytochrome C will release out, and leakage of cytochrome C, this leakage of cytochrome C, when cytochrome C release out, what happens? This cytochrome C, it binds to APAF, that is apoptosis activating factor 1. So now when cytochrome C releases out, firstly the sensor BS3 proteins are activated, BAD, VIM, BID, NOXA and PUMA, and those will help leakage of cytochrome C. Cytochrome C will leak out. How cytochrome C will leak out? Same thing, MPTP will activated, and obviously their leaky channel will be forming, and that will help in leakage of cytochrome C. So cytochrome C will leak out of those membranes. So that will result into activation of cytochrome C will bind to apoptosis activating factor 1. So they will form apoptosome. So apoptosomes are formed. So apoptosome are nothing but it is a combination of cytochrome C with apoptosis activating factor. Now this apoptosome will activate caspases 9. It will activate caspases. We know they are in the form of procaspases. So the procaspases 9 will be activated. So hence this is initiation activating cascade of caspases. So caspases initiation, caspases are activated, caspases 9, and cascade of caspases activates now. So the cascade of caspase activates now. So now this caspase of caspase activated, and this caspases was all about initiation caspase. Now execution caspase will also come in action. Before that, so there are something which inhibits apoptosis also. So what are the inhibition of apoptosis occur? So inhibition of apoptosis, there are inhibitors. So there are inhibitors of apoptosis inhibitor. So and apoptosis inhibitors like IAPS are called inhibitors of apoptosis. So this is about regulation of apoptosis. As I said, first thing, zymogens are the first regulator, physiological regulator, natural regulator of apoptosis. Okay, zymogens. And then again IAPs are also the regulators of apoptosis. So they inhibit apoptosis. Remember, inhibitors of apoptosis proteins, IAPs will inhibit, they are the physiological inhibitors. Remember, SMAC and diablo are inhibitors of IAP. So SMAC and diablo, they are inhibitors of IAP, they are inhibitors of IAP. So the best IAP inhibitors of apoptosis. IAP is nothing but inhibitors of apoptosis, and the best IAP inhibitor is nothing but the best IAP inhibitor is XIAP. So XIAP 10 I don't know that is 10 IAP or ever. XIAP is the best inhibitor of IAP. So best IAP, there are different IAPs, there are number of IAPs, but best one is XIAP. So XIAP is the best one. Now, and SMAC and diabol are antagonists to IAPs. So here now we can understand one thing, that BCL2 is helping in apoptosis. So remember, BCL2 is helping in apoptosis. BCL2 is anti-apoptosis, is not helping in apoptosis. So BCL2 is anti-apoptotic, and this inhibitors of BCL2, IAPs are apoptotic proteins, are inhibitors of this, and SMAC and diabol are inhibitors of IAPs. Remember this, and SMAC. And obviously survival signal factors like growth factors and cytokines. Whenever there is survival, as I have told already, whenever there are survival signals, so obviously the survival pathway will act, AKT pathway, PI3 AKT pathway and all this. So this survival signal pathway like AKT pathway, this is inhibiting what? They are inhibiting bad beam or sensor apoptotic factors. So pro-apoptotic factors or sensor apoptotic factors, they are inhibiting those. So whenever there is survival signal or growth factors and cytokines, then apoptosis doesn't occur. So whenever the survival signal is absent, apoptosis occur. And anti-apoptotic BCL family, BC12 family is also anti-apoptotic. So whenever there is BCL2 well functioning properly, apoptosis doesn't occur. But Now, inhibitor of apoptotic proteins. IAPs are inhibitors of apoptotic proteins. They are the physiological inhibitors. Okay, so this are this by this way you can do regulation. So now there is one more thing which is important: flip. So what is flip? So flip is flies inhibitory protein. So we'll talk about that. So let's first come to execution of apoptosis. So in execution of apoptosis, so this is intrinsic pathway. So we have talked about intrinsic pathway where there is activation of caspases 9. So now after that activation of caspases 9, what happens? The activation of pro-caspases 3 and 7 occurs. So 367 is executioner pro-caspases. So activation of pro-caspases 3 and 7 occurs, which activation will result in activated 3 and 7, and hence they will result in cleavage of substrate or apoptosis. So this is execution phase. So in execution phase, you remember caspases 3 and 7. So now here again this figure is saying same thing. XIP will inhibit caspases 9, so apoptosis will not occur. So apoptosis will not occur, and diblo and SAM, they will inhibit this. So here this SMAC and diblo and SMAC, they are inhibitor of IAPs. So they inhibit IAPs. So they will inhibit IAPs. And there is side flip. So flip will inhibit procaspase 8. So this is for flip is for extrinsic pathway procaspase 8. Remember, procaspase 8 or extrinsic pathway, in male it is 10, caspases 10. So in male it is caspases 10. In male it is caspases, male human being, it is caspases 10. Now the extrinsic pathway, obviously extrinsic pathway will be inhibited by flip. So extrinsic pathway will be inhibited by flip. Flip will inhibit extrinsic pathway. So whereas flip blocks the activation of initiator caspase 8 in this, XIAP can block both the initiation phase by inhibiting caspase 9. Here remember, XIAP is inhibiting the initiation phase also and the execution phase also, and the execution phase by blocking caspase 3 and 7 of cascade. So this is about execution for apoptosis. So once the initiator caspases are activated, they cleave and activate executioner caspases, mainly caspases 3, 6 and 7. So 367, remember 367 is executioner. 367 is executioner, and 8 and 9, 9, 8, 10, 9, 8, 9, 10, 8, 9, 10, they are initiator caspases. So the active executioner caspases then clips each other, and in this way an amplifying proteolytic cascade of caspase activation is started, and hence there will be proteolytic DNA degradation, we call it DNA breakage, and hence. So that was about intrinsic pathway. So the extrinsic pathway also occurs in same way. There is initiator phase and there is executioner phase. So in that extrinsic pathway we have a fast receptors. So death receptor, fast receptor, that is CD95. So this is called death receptor CD95, and it have the obviously the organism or any the noxious agent that will have a fast ligand, like T cell have fast ligand. So obviously T cell, when they bind to any microorganism, they bring to the fast ligand, they will bring at fast receptor. So they have fast ligand. So the fast ligand, they will bind together, and hence they bind together and they activate the adapter proteins. So FADD is adapter protein. FADD is nothing but it is an adapter protein, FADD. So this FADD is an adapter protein, and this adapter protein obviously will, with this together with fast ligand, fast and FADD, it form, we call it as DISC. So we call it as this DISC, death inducing signaling complex. So it is a death inducing signaling complex, DET, death inducing signaling complex. So it is a death inducing signaling complex. So is formed, and hence this FAD FAD with adapter proteins are activated, and these adapter proteins will, so this is FAD is adapter protein, remember this is an adapter protein. So this is activated by polymerization. So here fast, and here what is occurring? Polymerization. So this is activated by trimerization. Okay, trimerization is occurring in this. So this is a death domain. Fast is CD45 is death domain. TNF factor is also a death domain, and obviously they will activate pro-caspase 8 will be activated to caspase 8, and hence they will activate exudational caspase. Exudational caspases are same for both pathway. Exudational caspase is 367 for both pathway, and apoptosis will occur. So death receptors, they are member of TNF receptors superfamily containing death domains essential for delivering apoptotic signals, and death receptors, they are activated by natural ligands and TNF family. So obviously T cells and all, T cell they activates death receptors. This receptor like CD95 or CD95 or trial R1, that is TNF related apoptosis inducing ligand R1 and trial R2, they bind to the fast ligands. So intracellular adapter protein FAD is fast associated death protein domain, also known as MORT1 is activated, which in turn recruits the pro-caspase. So I have already explained this. So this picture will sum up the intrinsic pathway and the extrinsic pathway. Obviously whenever there is any kind of cell injury, and we are talking about cell injury, and those cell injury will obviously activate the BCL family sensor, BCL2 family sensors. That is, we know that BS3 protein, sensor proteins, bad beam, puma and noxa. So they will activate, and obviously this activation will result into activation of apoptotic factors, and those apoptotic factor backs back, and hence they will affect on mitochondria, and this will in the mitochondria cytochrome C will be releasing. Cytochrome C will combine with APAF, and then the initiator caspases will activate, and that will activate the exudational caspase, and that will break down the cytoskeleton and apoptotic body is formed. So this picture summarizes our apoptosis, both type of intrinsic pathway and extrinsic pathway method. Now finally we come towards the difference between this necrosis and apoptosis. So now by this you know that there will be loss of membrane in this, there will be membrane blebbing but no loss, swelling will be there, here shrinking will be the seen, end with total cell lysis, no vesicle formation, there will be complete lysis, here there will be fragmentation in apoptosis. Obviously the disintegration or swelling of organelles will occur here. Mitochondria becomes leaky due to pore formation involving protein of BCL2 family. So here there will be poorly leaky mitochondria, here will be swelling of the organelles. So necrosis, biochemical features, obviously in apoptosis ATP process will be present, and it will be not affected that much. Here loss of regulation of iron means no energy requirement. So here apoptosis energy is required, necrosis energy is not required. Random digestion, we have already wrote this, smearing pattern and ladder pattern. And post-lytic DNA fragmentation is late process in necrosis. This DNA fragmentation is the major process in this apoptosis. So obviously it affects group of cells, it affects individual cells, and it will be evoked by different disturbance. It can be by physiological, different hormonal problem, and also loss of growth signals or different direct injury, radiation injuries. Phagocytosis also occurs in this also, and by this in this also phagocytosis, but phagocytosis occurs more in apoptosis. No inflammatory response, significant inflammatory response is seen in this. So there are physiological changes, and these are about the what are the misfolding disorders which can occur in case of apoptosis, which I will talk about, like Alzheimer's disease with A beta peptide protein affected will be A beta peptide, like cystic fibrosis, CFTR, Tay-Sachs disease, hexosaminidase, and Creutzfeldt prion. So these all are protein misfolding disorders in which apoptosis occurs. This figure again sums up the intrinsic and extrinsic pathway which we have discussed already. So this ends our topic regarding the apoptosis and necrosis. So just remember apoptosis and necrosis, and we have summarized this. I hope you understood this topic very well. we are able to cover only necrosis and apoptosis today. So we'll talk about autophagy and other type of deaths in next coming class, and also we'll learn about different intracellular accumulations and calcifications which occurs in this, that is also cellular response. So the best way to study is read and come. So try to read this everything, read all these thing again, read these things, and then try to solve those questions which were given in our first slides. So try to solve these questions. Now I think you can solve these starting questions, and hope you get everything, and hope you are clear about all of these things, and this will help you. So like this, the first question: in irreversible cell injury, microscopic change seen is. So there will be no leakage of enzyme in this, mitochondrial density, cytoplasmic vacuoles, or pycnosis. So now you can basically see say what are the basic changes in reversible cell injury, and obviously cytoplasmic vacuole, hydropic vacuoles are the first thing which are seen in reversible cell injuries. Mitochondrial densities you can see, but that very hard, or that are irreversible cell injuries. Which of the following is an example of physiological hyperplasia? Now you can answer this question. Basically, physiological hyperplasia. Now this is a controversial question. Physiological hyperplasia. So which of the following is an example of physiological hyperplasia? Endometrium during pregnancy, liver regeneration after rejection, breast during pregnancy. So there everything can go in physiological hyperplasia, but basically which one is going more in this is endometrium during pregnancy. Now blebs are found in which type of injury? Basically reversible injury. What is the first sign of injury? So first sign of injury, that is sign. There are difference between sign and obviously this hallmark. So sign of injury is mitochondrial dysfunction is first sign of injury. So membrane damage is late in irreversible injury. So you can solve these questions now, and you can understand the concept very well, and hope you will be doing good. See you again in next class. Thank you.